I gave an overview of immune and cellular therapy in AML, so sort of the ABC of immunotherapy in AML. And let's start with A, antibody-drug conjugates, fusion proteins, and monoclonal antibodies. And just as a reminder, gemtuzumab, an ADC directed against CD33, is the only approved immunotherapy in AML. And there are others, ADCs and fusion proteins, that are currently in clinical development, and after showing safety in the relapsed refractory setting, have now actually entered the newly diagnosed space, so currently being applied as triplets in conjunction with Ven/Aza, giving really promising data. And I think, for example, Ludwig-Maximilians-University as also Tracoxifos have to be followed. Really interesting. We'll see where this will go. Then we have the monoclonal antibodies. Mainly, they have, some of them have actually achieved and reached phase 3 trials, but unfortunately, so far, none of them have shown a clinical benefit. So we've seen data from zabatolimab, an anti-TIM3 monoclonal antibody, which was applied in conjunction with AZA in high-risk MDS patients, but failed to show a benefit, although there was a prolongation in duration of response. But that program was terminated due to futility. And then we had magrolimab, the high-affinity anti-CD47 antibody, which was tested in the ENHANCE 2 and 3 trial, both randomized clinical trials recruiting internationally, and both of these were terminated early. The program closed due to futility, and one of the obstacles that contributed to this was toxicities like phagocytic anemia, and we'll see how other CD47 directed antibodies will evolve that are currently still running in phase 1-2 trials. Then B, for bispecifics, I summarized the early clinical trial data from phase 1-2, and I think across the board, targeting CD123, CD33 and CLL1. In most of these clinical trials, we've seen a high rate of cytokine release syndrome. So most of these trials implemented dose steps, making it really difficult to recruit patients and led to a protracted patient recruitment. And also it was contributed to the difficulty to achieve the final dose. And unfortunately, and across trials, what you can see is, although overall response rates went up to 50% in some of the trials, and CR/CRi rate up to 30%, none of these were sustained. So currently, things are trying to improve, and 4 aspects have been implemented in early clinical trials that are currently running. First of all, increased specificity against AML cells. So we have constructs that are now multi-targeting or targeting intracellular antigens to increase specificity. Second, the integration of positive co-stimulatory domain, either within the construct or applying a second bispecific antibody. And third, giving bispecifics in combination with, for example, Ven/Aza in our group has shown preclinically that you can thereby increase efficacy. And fourth, I think really important, applying bispecifics in low disease burden, preferentially in the MRD setting. And I'm really happy that some of these clinical trials are now recruiting for MRD positive patients. So we'll see if these strategies will advance and increase the success of bispecifics. And lastly, ABC. So the C is for CAR T cells. And here I introduce the 3 different strategies that are currently being followed in the context of AML. So first of all, we have CAR T cells as a bridge to transplant. So here the aim is to achieve morphologically leukemia-free status in the bone marrow, preferentially MRD negative, and then follow with a transplant. So the conditioning is eliminating all the CAR T cells, and then you have an allo salvage transplant. And several clinical trials have reported early results, and I think proof of concept has been shown. And sort of across these trials, about 30 to 50% of these patients actually achieved MRD negativity and could go on to allotransplant. However, we are missing long-term follow-up, and also we are missing biomarkers, and we don't understand which patients are actually responding, why they are responding, and vice versa. So the second strategy, really interesting and based on novel technologies, is using a stem cell edited graft followed by CAR T cells. So this was first shown preclinically with CD33 knockdown stem cells and then adoptive transfer of CD33 directed CAR T cells. So the hematopoietic system becomes invisible to the CAR T cells. And now actually first proof of concept has been shown clinically with patients that received CD33 deleted stem cell grafts, and then after reconstitution, so first of all showing this is feasible, that giving gemtuzumab, a CD33 directed ADC, did not lead to any cytopenias. And now with further advancement and CRISPR strategies, you can integrate non-synonymous mutations against the epitope that is actually recognized by the CAR T cells. And at least preclinically, that has been shown now for CD45, FLT3, 123, and c-KIT. And hopefully, clinical trials will start in 2025. And lastly, we have the selectivity enhanced CAR T cell approaches that are trying to do CAR T cells without an allogeneic stem cell transplantation through dual targeting CAR T cells or adopter CAR T cells. So a lot of technology evolving. I think we have to increase our biomarker program to better understand which patients are actually benefiting, and that applies for the monoclonal antibodies, for the bispecifics, as well as the CAR T cells. We need to harmonize this so that we can better compare between these trials. And I think in general, it is important that we apply these immunotherapy strategies in low disease burden, preferentially MRD. So this was my ABC of immunotherapy in AML. Thank you.